This lecture is continuation of neuroanatomy series. Today we are going to learn about action potential. As I said, the neuron is an excitable cell and it generates the impulses. Today we will see how these impulses are generated because whole the nervous system is based on these impulses. The nervous system works because of these impulses. So it is very important to know how these are generated. So this is neuron and this is cell body or soma. This is dendrites. From where the impulse reach to the neuron from another neuron, or the action potential can initiate in the neuron itself also. This is plasma membrane that forms the external boundary of neuronal cell. It is the site of initiation and conduction of the nerve impulse. The plasma membrane is composed of an inner and an outer layer of loosely arranged protein molecules, and both the layers are separated by a lipid layer. Lipid layer is made up of two rows of phospholipid molecules arranged so that their hydrophilic ends are in contact with each other and their polar ends are in contact with protein layers. Why I am focusing on this plasma membrane structure? Because it is very important to understand this structure to understand the action potential. This is called lipid bilayer because this is arranged in two layers of phospholipids. And this phospholipid molecule is made up of phosphoglycerol head and two fatty acids on the tail. These fatty acids are lipids, that's why they are hydrophobic, and this head is hydrophilic. This hydrophilic end of the phospholipid is charged, so it does not allow the charged ions to pass through. And the molecules that are not charged but are big enough to pass through the spaces between this lipid bilayer can also not pass the plasma membrane. The molecules that are not charged and are small enough to pass through the spaces between this lipid bilayer can go through the hydrophilic end but will not be allowed ultimately by hydrophobic ends, which are a sandwich between two hydrophilic ends, so they can also not pass the plasma membrane. So only small and hydrophobic molecules are able to diffuse across the phospholipid bilayer at a significant rate, like molecules of gases, oxygen, carbon dioxide, etc. Now some proteins are arranged on inner and outer layer, but some proteins span whole the length of the lipid bilayer, and these proteins provide the channels for the hydrophilic molecules and ions. Now the ions for us to focus here are potassium and sodium ions. In the resting state, potassium and sodium ions diffuse in the cell cytoplasm from the tissue fluid, and the permeability to the potassium ions is much greater than the permeability from the sodium ions. Thus, the passive influx of the potassium ions is much greater than the influx of sodium ions. Because on the resting stage, more ion channels of the potassium are open than the ion channels of the sodium. Now, a sodium potassium pump is also present inside the cell that pumps two potassiums in the cell and three sodiums out of the cell. Keep in mind that I told you less gates are open for sodium and more gates are open for the potassium that are different from sodium potassium pump. Sodium potassium pump is a active transport of sodium potassium and the gates are passive or facilitated transport. That means two mechanisms are active to keep less sodium inside and more potassium inside. But the sodium potassium pump is doing one more thing, that it is draining one positive charge out of the cell. This makes the scarcity of positive charge inside. Now inside the cell there are many proteins, and proteins are negatively charged molecules, and the DNA of the cell is also negatively charged. So the negative charge of the cell is greater than the positive charge, which makes the cell to be polarized and acquire a voltage, a negative voltage, which is difference of potential between intra and extracellular spaces. Now the intracellular space is more negative than the extracellular space. So the extracellular space is more positive than the intracellular space. You can consider it as a battery that is not connected with a circuit. So the flow of charge or the flow of electricity is not initiated yet. Resting membrane potential of the neuron is minus 70 millivolt. Means The neuron is minus 70 millivolt negative than the outer environment. And this is not a static equilibrium, this is a dynamic equilibrium. All the ions are coming to the cell and get back to outside. The sodium potassium pump has a major role to maintain this resting membrane potential. A neuron can be stimulated by mechanical, electrical, or chemical means. As soon as the neuron is stimulated, the membrane permeability for the sodium ions is increased. Means the more channels of sodium ions are open and a rapid influx of the sodium ions takes place because the concentration gradient of the sodium ions is from outside to inside. Now this decreases the negativity of resting membrane potential. This activity ceases in 5 milliseconds and then the voltage gated potassium channels open. Now the potassium ions start to flow from inside the cell to outside the cell because the concentration gradient of the potassium is from inside to outside. So now the resting membrane potential will get back to the normal. As sodium ions start coming inside the cell, the negativity of the cell lessens due to incoming positive charge and it becomes plus 40 at one time. In about 5 milliseconds, the sodium channels close and the potassium channels open. And then the rush of the potassium outside the cell makes the cell gradually less positive. And then zero and potassium keeps going out and cells start becoming negative. And at the equilibrium of minus 70, the resting membrane potential is achieved and the sodium and potassium channels get back to their original permeability, that was less for sodium and more for potassium. Let's see this chart. This chart is showing all what is happening in the cell. Minus 70 is resting membrane potential. At minus 70, some stimulus came, and if that stimulus was strong enough to open the ion channels for such a time that the voltage increases minus 55 volt, the action potential will happen. But if the stimulus is not strong enough to open the channels for such a time that the voltage increases minus 55 volt, but it increased in between minus 55 to minus 70, for example minus 60 or minus 65, then the action potential will not happen. Or the sodium came in the cell for that stimulus, it will be pumped out from the sodium potassium pump. So the threshold of action potential is minus 55 millivolt. Now let's see what is refractory period. Refractory period is a period between one action potential to next action potential. As you saw, the resting membrane potential was achieved by pumping potassium out of the cell and the sodium was in the cell, and that is not the actual proportion of the ions in the cell. The potassium is in more quantity and sodium is in less quantity. So this is achieved back by the sodium potassium pump, and that time where this concentration is achieved back, it is called refractory period. And in the refractory period, next action potential cannot be elicited. Now let's see how action potential is propagated. For example, action potential initiated in this region. This region depolarized and sodium channels open. After 5 milliseconds, sodium channel will be closed and the potassium channel will be open, and this region will start repolarizing. But the depolarization of this region affected this region and this region started depolarizing, and that's how the impulse will propagate throughout the neuron and that neuron will stimulate another neuron. I hope I am successful to clear this difficult topic. Till the next video, Khuda Hafiz.